Hi everyone, this is Chapter 10, the nervous system. For any of you that were in class on Tuesday, this beginning of this lecture will be a repeat, but it never hurts to repeat things. So this lecture is going to go through every slide that I have for Chapter 10, the whole nervous system, and it coincides with all of the pages that are assigned to you in the syllabus. So here we go, the whole Chapter 10. All right, so our nervous system is made up of three things. It's our brain, our spinal cord, and then basically the third thing is all of our nerves that leave those areas and travel out to our organs and our extremities. So the job of this system, I like to think of it as kind of like the U.S. Postal Service. It's a messenger or messenger system. It receives messages, so it gets input, and it also transmits messaging, meaning sending output out to the body on what to do. So along with the endocrine system, the nervous system coordinates the activities of the trillions of cells that we have in our body. And so these two systems are our controllers, the nervous system and the endocrine system. They perform similar functions in the body. However, they perform those functions in different ways. The nervous system uses electricity or electrical impulses to control the cells in our body. And the endocrine system uses hormones, basically chemicals that are in your bloodstream that are released from glands. So both of these systems, the nervous system and the endocrine system, they perform similar roles in our body. They just do them in different ways. So these two chapters are lumped together for Exam 4 because of this similarity in their roles. So the nervous system has many divisions as well as subdivisions, and I'll cover all the subdivisions in a little bit later in this lecture. But for right now, let's start with the primary division. Essentially, our nervous system can be divided into two umbrella systems. One is the central nervous system, and the other is the peripheral nervous system. So that's our first division. The central nervous system, or the CNS, is just our brain and our spinal cord, so those two organs. And then the peripheral nervous system is essentially everything else. So all of the nerves that leave the brain and spinal cord, and all of the sensory organs that we have throughout our body. So essentially, the peripheral nervous system is everything but the brain and the spinal cord. So there are two types of cells that make up the nervous system. First, we have neurons, which we'll talk about in a second. And then we also have neuroglia, which is what we're going to talk about right now. So neuroglia are also called glial cells, and they are the supportive cells of the nervous system. So glial cells do not send impulses like the neurons do. They basically protect the neurons and then glue them together, as well as perform various functions that enhance the performance of the neurons. So they're the supporters. The neuroglia are the supporter helper cells to the neurons. So for each neuron, there are approximately 50 glial cells supporting it. It's kind of crazy. And these glial cells come in 5 different types. And you'll see this on the slide here. So let's start with the first type of glial cells, the oligodendrocytes. So the oligodendrocyte cells, they make up the myelin sheath that covers all of the neurons in the brain and the spinal cord. So they are only found in the central nervous system, and they form the myelin. The second type of glial cells you see here is the ependymal cells. So the ependymal cells provide the inner lining of things such as the spinal cord and the cavities within the brain. So they're on the inside of everything. And these cells are responsible for secreting our cerebrospinal fluid, which is the fluid that actually runs through the whole central nervous system. Central nervous system does not have blood running through it. It only has cerebrospinal fluid, and that's produced by these ependymal cells. Our third type of glial cells is the microglia. So the microglia perform phagocytosis. If you can remember from earlier chapters, phagocytosis refers to cellular eating. So microglia cells are like the house cleaners, and they engulf basically any microorganisms and debris that is unwanted or old. So it just kind of gets rid of all of the junk. Next you have astrocytes. So astrocytes, also only found in the central nervous system. These cells attach neurons to the blood vessels and help form what's called the blood-brain barrier. So like I said earlier, the central nervous system does not have blood flowing through it, but capillaries are right outside of the central nervous system. And these astrocytes form the barrier between the central nervous system and those capillaries, which is called the blood-brain barrier. So this barrier exists to protect the brain from foreign substances, but it also is real picky, and it will allow only the needed things through to go into the brain and the spinal cord. So those needed things would be like oxygen and glucose, as well as other nutrients. And the last glial cell you see is the Schwann cells. So the Schwann cells, these are the only ones up here on this list that live in the peripheral nervous system. The rest of them belong in the central nervous system. So the Schwann cells perform the same role as the oligodendrocytes, just on the neurons that are in the peripheral nervous system. If you remember, the oligodendrocytes and the Schwann cells, they form the myelin sheath that wraps around the axons of the neurons in both of those divisions of the nervous system. So let's talk about these astrocytes for a minute. So these astrocytes, you can see the image on the slide. They're the most numerous of all of the glial cells. They're quite pervasive throughout the brain. And they're shaped like a star, which is why they get the name astro. And they have these tiny little feet at the end of each projection of that star shape. And these feet latch onto capillaries and other neurons to basically feed the neurons the glucose from the bloodstream and any oxygen and nutrients that it needs. So their attachment forms this blood-brain barrier. And like I mentioned earlier, what protects the brain by only allowing, the blood-brain barrier protects the brain by only allowing the small necessary nutrients through, and it does not allow anything that could be damaging. So this barrier also prevents most medications from getting through to the brain as well, which is a good thing sometimes, but it makes it hard to treat disorders of the brain, such as brain cancer, because there's a barrier preventing things that are either injected or taken orally, those kinds of medications, to even reach the brain. So our brain is so important that we have a whole mechanism that is basically the stop of everything before it gets to the brain. So this barrier only allows the approved things through and not the unimproved or the not improved things. So now let's switch to the other cell that is found in the nervous system. It's the neurons. So the neurons are the actual messengers. They handle all of the communication, or aka the electrical impulses, that send messages throughout the whole body. So there's 3 types of neurons in our body. First, you have sensory neurons, also called afferent neurons. So these neurons detect stimuli such as touch or pressure or heat or cold or chemicals, anything that our sensory organs sense from our environment. And they transmit this information about that stimuli to the central nervous system. So I like to think of the afferent neurons, the messaging traveling in the direction from the peripheral nervous system to the central nervous system. Next, you have interneurons. So interneurons are only found in the central nervous system, and they connect the incoming sensory pathways with the outgoing motor pathways. So they are like the transfer station for those afferent neurons to the efferent neurons. And then third, you have the motor or efferent neurons. And these neurons relay messages from the brain and the spinal cord to the rest of our body. So basically, a motor neuron is considered an efferent neuron. If you can remember from the muscle chapter, we talked about what a motor neuron does, how it causes a contraction. And so that is an efferent neuron. So let's visualize this process. Our hand is about to touch a hot pan, and when it happens, sensory organs in our skin send a message through the afferent neurons that this thing that we're about to touch is hot. So then the interneurons in the central nervous system, they get that message, they make a decision with the specific part of the brain for that sensation, and then they send a message to move our hand out through the efferent neurons. Now, this process happens at the speed of light, which is really good, obviously, but it goes through this pathway all the time. Afferent information is sent to the interneurons, that then is message is sent back out through the efferent neurons. So the structure of a neuron should be a review to us, right? So you have the cell body, or the soma. This is where the nucleus resides. And you have the dendrites, and that's what receives the impulse surrounding the cell body. And then you have the axon, which is where the impulse leaves the cell body. But let's add a few more structures here. So the axon has many, but not all neurons the axons of many neurons, but not all neurons, are encased in what's called a myelin sheath. And this is what insulates the axon. So the myelin sheath consists mostly of lipids, and in the peripheral nervous system, this myelin sheath is formed of Schwann cells. And this is what you're looking at on this slide. This is a peripheral nervous system myelinated neuron. So the oligodendrocytes form the myelin sheath in the central nervous system. However, they're arranged a little bit differently. So the spaces between the myelin sheath, those are called the nodes of Ranvier. And these are evenly spaced between each other. And then at the very end of the neuron, or the axon, is the synaptic knob. And so this is the terminal end of the axon, which is where all of those vesicles live that contain the neurotransmitters. And if you can remember, the neurotransmitter for a motor neuron is acetylcholine. So the vesicles that contain the acetylcholine, they live in the synaptic knobs of the axon. Some neurons will be myelinated and some won't have myelin. And the reason for this difference is mainly the need for speed and the impulse transmission. So myelin makes that impulse transmission way faster. So unmyelinated neurons will conduct a lot slower. For example, the neurons that control your digestion, the neurons that control your breathing or your kidney function, they are all unmyelinated. There's no need for speed for those functions. But muscle cells, they need quick impulses to make us contract immediately, right? So motor neurons will be myelinated. For those that are myelinated in the peripheral nervous system, the glial cell, just as a reminder, is the Schwann cell. And the myelin sheath in the central nervous system, the cell that makes that up, is the oligodendrocyte. So the way these two cells work differ. A Schwann cell will wrap itself around the axon of one neuron, laying down multiple layers of the cell membrane. And you can see this on the image on the left. So the inside layers form the myelin sheath, but the nucleus and most of the cytoplasm of the Schwann cell kind of gets squeezed to the outermost layer of this wrap. And this outermost layer has a name, and it's called the neurolemma. Because the myelin on this neuron has lots of neurolemmas, this is where the nucleus is, and that means that this myelin can actually regenerate if it gets damaged because that nucleus is present. So this is why we can regrow and restore function in our neurons in the peripheral nervous system. However, in the central nervous system, the oligodendrocyte wraps itself quite a bit differently. So one oligodendrocyte will wrap projections of itself around the whole axon of several neurons nearby. And this causes the nucleus of the cell to be located away from the myelin sheath, and therefore there is no neurolemma around that axon. And because this nucleus is far away, this prevents injured central nervous system neurons from regenerating. This is why injuries to the spinal cord and the brain are sometimes considered permanent. So an interesting fact, myelination, or the development of our myelin, begins during the 14th week of fetal development. And we actually continue to develop our myelin until late adolescence. So during pregnancy, a mother's nutrition and health is imperative in the healthy development of myelin in her baby. And the same goes for the nutrition of children as well as adolescents. So, like I mentioned before, neurons transmit messages via electricity or electrical impulses. At the molecular level, electrical impulses are basically the result of positively and negatively charged ions flowing from one point to another. And if you can remember from chapter 2, an ion is an atom that has either lost or gained an electron, and therefore it causes it to have a positive or a negative electrical charge rather than a neutral charge. So that's why you end up with positive ions and negative ions. So in the body, these oppositely charged ions, they'll travel into and out of the neuron across the neuron's membrane. Whenever one side of the membrane has predominantly one charge and the other side of the membrane has the opposite charge, the potential exists for them to move towards one another, because opposites attract, right? So this is called membrane potential. So this can also be called polarization. So polarization is what causes the membrane to have potential for these ions to travel across the membrane. So when a neuron is at rest, meaning it is not conducting an electrical signal, the interior has a negative electrical charge, and then the exterior has a positive charge. You can see this on the slide. Also, the interior contains an abundance of potassium ions, and the exterior is quite rich with sodium ions. You don't see this on this slide, but imagine a bunch of potassium on the inside and a bunch of sodium on the outside. So both sodium and potassium are positively charged, but also within the interior are large negatively charged proteins and negatively charged nucleic acids that basically overrule the potassium that is positive and give the interior an overall negative charge. So as we walk through the steps of impulse conduction, I want you to pay attention to two things. One, what is the charge on the sides of the membrane along the axon? And two, where are the potassium and where are the sodium ions located? Because these are the things that change within each step of impulse conduction. So to start out, because the membrane's really permeable, a certain amount of sodium and potassium ions leak across the membrane all the time, even when an impulse is not happening. However, the sodium-potassium pump, which we talked about in Chapter 3, is constantly working to restore those ions to their appropriate size. And if you can remember from Chapter 3, the sodium-potassium pump works like this. It sends 3 sodium ions out at the same time it takes 2 potassium in. This pump is located right inside, kind of embedded in the membrane, and just is a gate that allows sodium out and potassium in. So this state right now of being inactive and polarized is called resting potential. So the neuron is resting, but it has potential to react if a stimulus comes along. So this is essentially step 1. So in step 2, let's say a stimulus does come along. The stimulus could be a sensation of chemicals or heat or mechanical pressure. It could be anything. And what this does is this causes sodium channels on the neuron's membrane to open in that region, right at the connection of the axon to the cell body. And this allows sodium from the outside the membrane to rush into the cell. Now these channels are different than the sodium-potassium pump, which is what pushes sodium out and potassium in. So these channels are different. They're embedded in the membrane as well, but these channels, they let sodium only in and they let it in passively. So this addition of the positively charged ions, it changes the charge of this beginning region of the axon's interior from negative to positive. And then the membrane begins to do what's called depolarize as the charge begins to change. So this is step 2, step of depolarization. So as a review, depolarization happens when a stimulus comes along and it causes sodium channels to open, and then sodium rushes into the membrane of the cell. So if you guys remember from the motor neuron discussion, that motor neurons respond in an all or nothing kind of way. And this type of response, this all or nothing, it goes for all neurons, not just those motor neurons. And so what I'm referring to is when a neuron reaches, or the stimulus reaches threshold. Nothing will happen until it reaches threshold, and nothing greater happens if the stimulus is greater than threshold. So that's what's happening here. So this depolarization, it has to reach a threshold level before anything else continues to happen. So basically, enough sodium needs to flux into the neuron to meet this threshold of depolarization. But once it does, though, action potential happens. The adjacent channels next to that original channels of sodium, they open up and they allow even more sodium into the neuron. And this continues all the way down the axon as one segment stimulates the next, that stimulates the next, that stimulates the next, and so on. So this is called action potential. This is when the nerve impulse actually gets sent. The neuron is in action. So action potential is step 3. Now, all of this sudden influx of sodium, it causes the interior to become positively charged. And this triggers potassium channels to open up behind these sodium channels, and it sends sodium out of the cell. So this is done to make the interior negative again, because that's where it wants to be. Soon after the potassium begins to exit, the sodium channels from earlier shut to prevent any more from flowing into the cell. So essentially, this repolarizes the cell. However, if you notice, the ions are flip-flopped in their position. You end up with potassium more on the outside and sodium more on the inside. So this is the state of repolarization in step 4. You get the repolarization of the negative charge and the positive charge in the appropriate place. However, the ions, the potassium and sodium, are flopped. So as long as these sodium and potassium ions are on the wrong sides of the membrane, the neuron won't respond to a new stimulus, even though the membrane is polarized. But the neuron wants to be in its resting potential state again very soon, so the sodium-potassium pump goes to work. And this returns the sodium to the outside, and it brings the potassium back inside. So this is called the refractory period, step 5. So when this period is completed, the neuron is actually polarized and the ions are in their happy place. And this restores resting potential and the neuron is ready to receive another stimulus. So every movement, every process in your body, every thought, all of these things require several neurons to be doing this impulse conduction over and over and over again. And that means that this sodium-potassium pump, well, you got many of them per neuron, they're using a ton of ATP to do their job. And that means that the neuron's system is consuming a lot of fuel to produce this ATP. And the fuel that these neurons consume is glucose. So glucose is the only fuel that the neurons can use, and they need a lot of it to produce this ATP to then energize the sodium-potassium pump. And so it's estimated that about 120 grams of carbohydrate, which ends up being glucose in your body, actually goes just to fuel this system per day per person. So that was the system of impulse transmission for unmyelinated neurons. Myelinated neurons work a little differently though. The myelin blocks the free movement of ions across the cell membrane, so the only place that ion exchange can actually occur is at the nodes of Ranvier. So the action potential, or the electrical changes that occur in the nodes of Ranvier, and the current actually flows under the myelin sheath to the next node. And this triggers the next channel. Because the action potentials only occur at the nodes, the impulse seems to leap from node to node. And this type of signal conduction is called saltatory conduction. The distance between the nodes in myelinated nerves is way larger than the distance between the sodium channels in the unmyelinated nerves. Therefore, the impulse travels further quicker in the myelinated neurons, making these much faster neurons for impulse conduction. Feel free to watch the animation. You can see the link on this slide to kind of show you how this whole process works. So to keep the messages going, impulses must move from one neuron to the next, passing through what's called a synapse. So a synapse is the gap or the built-in space between the end of one neuron and the beginning of the next. So here are the steps on how this works. When an action potential reaches a synaptic knob of the axon, the membrane depolarizes. And this causes another ion channel to open up in the synaptic knob. And this ion channel is a calcium ion channel. So the channel opens and allows calcium to then enter the neuron. The infusion of calcium causes those vesicles that contain the neurotransmitters to then migrate and bind to the cell wall, and then they open up and release their store of neurotransmitters into that space or the synapse. The neurotransmitters will then travel across the synapse to the next neuron. The neuron that's sending the impulse is called the presynaptic neuron, and the one that's receiving the impulse is called the postsynaptic neuron. So these neurotransmitters, they bind to their specific receptor on the postsynaptic membrane. The neurotransmitter can cause two actions though. It can determine if the impulse continues, which is called excitation, or whether the impulse stops, called inhibition. If the impulse is inhibitory, potassium channels will open on the postsynaptic neuron and the impulse will actually stop. However, if the neurotransmitter is excitatory, as shown here in the slide, sodium channels open and the membrane starts to depolarize, and then eventually the impulse continues just like it did before. And last, the receptor releases the neurotransmitter, after which it is reabsorbed by the synaptic knobs and recycled, or it's destroyed by enzymes. Again, you can see a link on this slide if you'd like to click it and view the animation of this process. On this slide, there is a YouTube video of basically all the steps of impulse conduction all the way through to when a presynaptic neuron sends a signal to a postsynaptic neuron. So feel free to watch this whole video. It's in 3D animation, so it's pretty good to view, to watch this whole process kind of linked together. All right, so now let's kind of switch gears and let's talk about these divisions of the nervous system. As I said earlier, the two primary divisions that we have are the central nervous system, or the CNS, and that consists of the brain and the spinal cord. And then we've got the peripheral nervous system, which consists of all of the nerve networks that leave the brain and spinal cord. All right, let's dive a little deeper into the rest of these subdivisions. If you notice, on the left side of the screen, you have the central nervous system, and it subdivides into the brain and spinal cord. And then that is basically it for all of the central nervous system divisions. But if you look at the peripheral nervous system, you'll see that it subdivides, then subdivides again, and subdivides again. So let's take a deep dive into the subdivisions that exist in just the peripheral nervous system, not the central nervous system. So subdivision 1, under the peripheral nervous system, you have the afferent division and the efferent division. So remember these words from earlier. I want you to think of the afferent division as your inbox. So these are the neurons that receive sensory messages from the nerve endings in our organs, and then they head towards the central nervous system, your central command center. Then think of the efferent division as your outbox. So this consists of all the motor neurons that then take messages from the central nervous system and deliver them out to the target organs. So these two subdivisions basically specify the direction of travel for all of the messages. The afferent division comes in and the efferent division heads out. So now let's subdivide again. So within just the afferent division, there are two additional divisions called the somatic and the visceral sensory. Somatic sensory neurons are messages that come specifically from your structural and movement organs, such as your skin, your bones, your joints, and your muscles. And the visceral sensory neurons are messages that come specifically from your gut organs and your chest organs, such as your heart, your lungs, your stomach, your bladder, your kidneys, all of those things that are piled into your thoracic and your abdominal cavities. Now, within the efferent division, there are also two subdivisions. These are called somatic and autonomic motor. Somatic motor are the neurons that take messages to those structural and movement organs that we referred to in the somatic sensory. So these allow voluntary control of your muscles, which actually moves your body, right? And then the autonomic motor are the neurons that take messages to all of your organs in your gut and your chest and control all of those functions. So the autonomic motor functions are involuntary. They are the things like breathing and your heart rate, and your kidney filtration rate, and the things that happen but you don't have to think about it. Those things happen in the background. So now let's subdivide a third time. And this final division happens only within the autonomic motor system. So there are two divisions here. One that are messages for arousal of the body, and one that are messages for calming of the body. Sympathetic are messages of arousal, and parasympathetic are messages of calming. An example would be increasing your heart rate and then decreasing your heart rate. So these messages are both autonomic motor messages, but one is arousal, increasing your heart rate, which would fall under the sympathetic division, and then one is calming, such as decreasing your heart rate, which would fall under the parasympathetic division. So that was all of the divisions. Now let's take a look at the anatomy of the spinal cord. So if you remember in the bones chapter, there was a big column of spaces where the vertebrae stacked up on each other. It was called the vertebral foramen, or the vertebral column. And that is the whole column that your spinal cord runs through within your spine. So the spinal cord relays messages from the brain to the body, and then from the body back to the brain. So it extends from the base of the brain all the way until the first lumbar vertebra, or L1, and then the cord begins to separate into thinner nerve roots called the cauda equina. So it gets its name because of how it resembles a horse's tail. So caudal refers to rear, and equine refers to horse. Now, branching off the sides of the spinal cord are 31 pairs of spinal nerves. They are divided into regions that coincide with the regions of the vertebrae. So nerves from the cervical region talk to the areas of your chest, of your head, your neck, shoulders, arms, hands, and even your diaphragm. And nerves from the thoracic region, they talk with the intercostal muscles of the rib cage, the ones that help you breathe, as well as the abdominal muscles and the back muscles, and a good bit of your gut organs. And the lumbar spinal nerves, they innervate the lower abdominal wall and some parts of the upper thighs. And nerves from the sacral region talk with the rest of the thighs, the buttocks, the muscles of the skin and the legs and the feet, and the genital regions. So this is a cross-sectional view of one vertebrae and a cut part of the spinal cord. So imagine we're looking down onto the cord from here. So within the spinal cord, you have two types of nervous tissue. You've got white matter and you've got gray matter. The gray matter is in this H shape, kind of a central formation, and the white matter surrounds the outside of the gray matter. The gray matter appears gray because there's no myelin here. It contains mostly the cell bodies of the motor neurons and the interneurons. The H-shaped mass is divided into two sets of horns, the posterior or dorsal horns and the ventral or anterior horns. So can you recognize which direction posterior and anterior are because of the vertebrae? Now, white matter appears white because of its abundance of myelin. It contains bundles of axons called tracts, and that carries impulses from one part of the nervous system to another. The tiny central canal that you can barely see on the slide, it runs the length of the spinal cord through the gray matter and carries cerebral spinal fluid through the spinal cord. Lining this central canal are one of those glial cells that produce that cerebral spinal fluid. And those cells, if you can remember, are the ependymal cells. And now there's a space between the outer covering of the spinal cord and the vertebrae around it, and this is called the epidural space. So it's mostly a cushioning layer of fat, but it also has blood vessels and connective tissue throughout it. And these are where the blood vessels run right up next to the spinal cord. So remember that the vertebrae are each connected to each other by an intervertebral disc. And that disc sits on that vertebral body of the bone, and it separates those vertebrae from each other. So this disc provides a gap between each vertebrae, which allows space for those spinal nerves to then exit the sides of the spinal cord. So these spinal nerves that exit through the sides of the gaps, this is how the messages leave the central nervous system and go out to the periphery. So each branch begins with a dorsal and a ventral root, and you can see the two there on the slide. And I want you to think of these two roots as the inbox and the outbox of transmission. The dorsal root carries information into the spinal cord, and the ventral root carries information out of the spinal cord. Within the dorsal roots are these little knot-like structures called ganglion. Ganglion are a collection of the cell bodies of a bunch of neurons. So these two roots join but contain mixed neurons in them. And they contain sensory and motor, so your inbox and your outbox, after this junction. So the impulse transmission happens both ways after that ventral and dorsal roots join. So this fusion, where the mixed nerves are, it results in what is called, in one group, a spinal nerve. So protecting the spinal cord is the vertebrae, of course, the bone. But also you have three layers of fibrous connective tissue surrounding it called meninges. So just like every other organ which we've learned about, there are tons of layers. So meninges is the name for the spinal layers. So the pia mater, the innermost layer, This is a transparent membrane that clings to the surface of the brain and the surface of the spinal cord. So this is actually where the blood vessels are contained. The arachnoid mater lies just outside the pia mater, and it's a delicate layer resembling kind of like a cobweb, hence the name arachnoid. The dura mater is the tough outer layer on its exterior. So there is a space between the arachnoid and the pia mater, and it's called the subarachnoid space. And this is where you also have cerebrospinal fluid residing. So what is an actual nerve? A nerve is a bundle of neurons, mainly the axons of the neurons, that are encased in connective tissue. So the number of nerve fibers contained in a single nerve varies to just a few to approximately 1 million. So kind of like our muscles tear down into smaller units, these bundles of nerves are called fascicles. So the fascicles are also individually wrapped in dense connective tissue and also contain blood vessels right up against them. So most of the time, these nerves contain both sensory and motor neurons, or the fibers, aka the afferent and the efferent. And these are called mixed nerves. And obviously they can transmit signals in both directions, to the central nervous system and away from the central nervous system. However, there are a few nerves, such as the optic nerve, that are only sensory nerves and only contain the sensory or afferent neurons. So they carry sensations toward the spinal cord only. And also there are a few nerves that are just motor nerves, and they contain only motor or efferent neurons and carry messages to the muscles and the glands. So most of them in our body, though, are mixed. So let's step away from anatomy, and now let's just think about function, or the physiology of our nervous system. So let's start with reflexes. So typically, sensory or afferent neurons, they send messages to the spinal cord, and those messages travel up to the brain for a decision to be made about them. But we also have these sensory and motor pathways that totally bypass the brain, and they arc or switch from sensory to motor in the spinal cord instead. For instance, you will sense something from an extremity like your hand, or viscerally from an organ, and this message goes to the spinal cord, but in a reflex, it then goes straight to the motor neuron as a response without going all the way up to the brain. So this bypass is called the reflex arc. It happens, and we aren't even aware of it happening until it's over. So there are two types of reflexes: autonomic or visceral, and then somatic. So what do you think the difference is? So autonomic reflexes relate to our internals, basically, our glands, our organs, the pupils of our eyes, peristalsis of our gut, things like that. And somatic reflexes relate to our movements. So an example of our somatic reflex is the patellar reflex. This is what you can see on the slide. So who remembers when their doctor hit the hammer on their knee and it made your leg kick out just on its own? So this is a test for a doctor to assess this reflex function in our nervous system. So this slide shows the example of the patellar somatic reflex. Here's how this goes. So somatic receptors located in the patellar tendon, they detect a sensation. When the hammer hits the tendon, it stretches fibers of the thigh muscle. Then two afferent sensory neurons send the signal directly to the spinal cord. And 3, the impulse immediately passes to a motor neuron in a reflex arc and bypasses the brain completely. 4, the motor neuron initiates the impulse back to the muscle, causing it to contract, producing a slight kick in your lower leg. And if you can remember back when this happened in the doctor's office, you didn't think about your leg kicking out, it just did it on its own. So that's an example of a somatic reflex. Now, a visceral reflex or an autonomic reflex arc, the same thing happens, but typically the brainstem area and not the spinal cord, which is just basically the top of the spinal cord. So somatic reflexes cause movement, but the visceral reflex causes internal things usually related to homeostasis to happen. The autonomic nervous system asserts control over internal functions through these visceral reflexes. So they will affect the internal organs rather than the muscles. All visceral reflexes follow similar steps. So this illustration shows the visceral reflex arc responsible for the regulation of blood pressure. So let's walk through these steps. So 1, receptors detect a change in the body's condition. Here, pressure receptors in the carotid artery, called baroreceptors, they detect a rise in blood pressure. 2, afferent neurons transmit that information about this change to the central nervous system. So in this example, the glossopharyngeal nerve relays the information to the medulla oblongata in the brainstem. Then 3, the medulla oblongata processes the information and it transmits a signal along an efferent nerve. Again, in this example, the vagus nerve sends a signal to the heart's pacemaker to slow its roll. And then 4, the effector organ, in this case the heart, receives that message and responds by slowing its rate, and therefore your blood pressure drops. So if you think back to our divisions chart, the somatic pathway encompasses the somatic sensory and the somatic motor divisions. The autonomic pathway encompasses the visceral sensory and the autonomic motor divisions. So if you combine the sensory and motor functions together, that is what the pathway is. So somatic is related to movements and autonomic is related to internal functions. So these somatic and autonomic pathways differ in how the neurons are actually arranged though. So on the slide, we have an example of how the somatic pathway is arranged. So let's look at this. So say a sensation comes in from a receptor in your periphery, and that message gets sent to the spinal cord or the brain. Now the neuron's cell body, the big part of the neuron, it actually lies within the spinal cord or the brain. And then get this, its single myelinated axon extends out of the spinal cord or brain all the way from that place in the central nervous system to the skeletal muscle. So at the target muscle, the neurotransmitter acetylcholine is released to cause that muscle contraction. So we have one motor neuron involved in the somatic pathway. It's myelinated and it secretes acetylcholine. So here's the difference that you will see in the autonomic pathway. So the cell body of the autonomic motor neuron resides in the brain of the spinal cord, just like the other one, and its myelinated axon extends out of the spinal nerve to the ganglion outside the central nervous system. So this myelinated neuron is called the preganglionic neuron. So remember, a ganglion is just a place in a nerve that houses a ton of cell bodies. Now, in the ganglion, it synapses with a second neuron, the postganglionic neuron. So the neurotransmitter used at this ganglion synapse is also acetylcholine. The axon of the second neuron, the postganglionic neuron, is actually unmyelinated. You can see this on the slide. So it extends to the target organ in the chest or in the gut, wherever it's going to. And the neurotransmitter released here varies quite a bit. So if the purpose of the neurotransmitter is arousal, sympathetic neurons will release the neurotransmitter called norepinephrine. If the purpose of this signal is calming, parasympathetic fibers release acetylcholine. So the difference is that the autonomic pathway requires two neurons. The first one is myelinated, the second one is unmyelinated, and it also differs by having two neurotransmitters, acetylcholine in the ganglion and norepinephrine or acetylcholine at the target organ. So why are there two neurotransmitters? Well, one is excitatory and one is calming. So this is basically the subdivisions of the autonomic nervous system back on a previous slide. So the sympathetic division prepares the body for activity, and the parasympathetic division calms the body down. So have you ever heard of our fight or flight reaction? This is where the sympathetic division is called on in times of stress or anger or fear. So say we sense something lurking around the corner of a dark, scary room. Our heart beats faster, our palms get sweaty, our breathing gets faster, and our pupils dilated. Our body is preparing to fight off this monster or bolt away as fast as we can. So this is the adrenaline that we feel from the sympathetic nervous system. So epinephrine and adrenaline are the same thing. And norepinephrine and noradrenaline are the same thing. These two chemical substances are both excitatory, but they have different receptors on the cells that they talk to. They can also both be a neurotransmitter and both be a hormone. So it's just called one or the other, whether they are released by a neuron or they're released by a gland. So both the sympathetic and the parasympathetic divisions work at the same time. And they provide a background level of activity called autonomic tone. So this balance, the balance though on which one is more dominant at the time constantly changes depending on the body's need. So if the body needs to rest and digest, the parasympathetic tone will be greater. And if the body needs to run away from a monster, then the sympathetic tone will be more dominant. So these two divisions also differ on where the neurons originate. Remember that the sympathetic and the parasympathetic are both autonomic, so there are two neurons in this pathway. The sympathetic preganglionic neurons begin within the spinal cord of the thoracic and the lumbar regions. So this picture depicts one side of the body, but understand that this happens on both sides of the spinal cord. So cell bodies in the spinal cord reach their myelinated axons to sympathetic ganglia just outside the spinal cord, really close. These preganglionic neurons are really short. Not all preganglionic neurons synapse in the first ganglion they encounter. Some actually travel up or down the ganglion chain to synapse with other ganglia at different levels. Others pass through the first ganglion to synapse with another ganglion a short distance away. They can basically go to any postganglionic neuron, which is how our fight or flight response is so immediate. The unmyelinated postganglionic fiber leaves the ganglia and extends to the target organs. Postganglionic fibers tend to be really long. Preganglionic fibers are short and depart from the thoracic and lumbar regions. The adrenal glands, which sit right on top of our kidneys, they play a large role in the sympathetic nervous system. So what hormone do you think the adrenal glands produce? It's adrenaline. And it's a hormone because it's coming from a gland. You can also call adrenaline epinephrine. So sympathetic preganglionic fibers, they pass through the outer cortex of the adrenal glands and they terminate in the center called the adrenal medulla. So this is where a mixture of epinephrine and norepinephrine is secreted into the bloodstream. Both of these hormones help prolong that sympathetic response, which is why it lasts for so long. Okay, let's switch gears and talk about our calming autonomic system, the parasympathetic division. So this is your rest and digest control. Unlike the sympathetic division that arises from the thoracic and the lumbar regions of the spinal cord, the neurons of the parasympathetic system arises from the brain and the sacral regions of the spinal cord. The ganglia of the parasympathetic division reside in or near the target organ instead of just outside the spinal cord. So this makes the preganglionic fibers very long and the postganglionic fibers very short, opposite of the sympathetic division. Because the ganglia are more widely dispersed, the parasympathetic division produces a more localized response than that of the sympathetic division. This means that it can just control the stomach and not affect the other organs if it wants to. So here is a helpful table that describes the key differences between the sympathetic and the parasympathetic divisions of the autonomic nervous system. So under sympathetic, it originates in the thoracolumbar region of the spinal cord. The ganglia lie in a chain alongside the spinal cord not far away. And that makes the preganglionic neuron short and the postganglionic neuron very long. And it employs mostly norepinephrine as the neurotransmitter, only occasionally does it use acetylcholine. And this produces a widespread generalized effect across the body. Now the parasympathetic, it originates in the craniosacral regions. And the ganglia lie in or near the target organs, which cause this long preganglionic neuron and a very short postganglionic neuron. Parasympathetic employs acetylcholine as a neurotransmitter primarily, and it produces a local effect rather than a systemic effect. So check out page 193 in your book. This is a list of all the physiological changes that occur different between the two divisions. Memorize those things. All right, let's now talk about those little chemicals that are released from the neurons that promote those differing functions of excitatory and calming. These are the neurotransmitters. So the neurotransmitter released and the type of receptor on the target cell determines these specific functions. So the autonomic nervous system uses two different neurotransmitters, acetylcholine and norepinephrine. Fibers that secrete acetylcholine are called cholinergic fibers, and fibers that secrete norepinephrine are called adrenergic fibers. Cholinergic sounds like acetylcholine. Adrenergic sounds like adrenaline, which is also norepinephrine. So the effects of the sympathetic division last longer than those effects of the parasympathetic division because acetylcholine is disposed of more quickly after the release than norepinephrine. So let's look at the differences of these fibers. Cholinergic fibers are typically preganglionic fibers of the sympathetic and the parasympathetic division. They're also the postganglionic fibers of just the parasympathetic division, and they secrete acetylcholine. Now, adrenergic fibers include most postganglionic fibers of the sympathetic division, and they secrete norepinephrine. So just like there are two neurotransmitters, cholinergic and adrenergic, there are two types of receptors for each of those fibers, just to confuse you more. So these two types of cholinergic receptors are called nicotinic and muscarinic. The different types of receptors determine the effect produced by the neurotransmitter. So if acetylcholine in a cholinergic fiber binds to a nicotinic receptor, it will create an excited response. But if acetylcholine binds to a muscarinic receptor, it will produce a variable response, which means it could go either way, excited or inhibited. An example of this is when acetylcholine stimulates the smooth muscle in our intestines, whereby inhibiting cardiac muscle during digestion. So this is a parasympathetic rest and digest function, but digestion obviously requires more activation of some organs. So acetylcholine is released for both functions, but these opposite effects occur not because of the neurotransmitter, but because of the different receptor. So the last slide was cholinergic receptors. So now let's look at adrenergic receptors. So norepinephrine is the adrenergic neurotransmitter, and there are two types of receptors that norepinephrine can bind to. You have alpha and you have beta. The difference is alpha causes an excited response and beta causes an inhibitory response. For example, the binding of norepinephrine to alpha adrenergic receptors in blood vessels causes the blood vessels to constrict. The binding of norepinephrine to beta adrenergic receptors in the heart and the skeletal muscles causes the vessels to dilate. So in the fight or flight response, you want blood to go to some places more than blood to go to other places less. So digestion will not help you survive the monster attack, but running away will. So vessels constrict in the unnecessary digestive organs during fight or flight, but dilation happens in the necessary organs such as the heart and the muscles to fuel you for running away. All right, well that concludes this lecture. So please go back and listen to, flip through, watch any animations just to make sure you've got it all straight. And please send me any emails with any questions that you may have, or if you need clarification for anything. So hopefully you've enjoyed this lecture. Okay, talk to you soon.